Hello, my name is Mikhail or Misha Kolonin. I'm at the Institute of Molecular Medicine at UT Health, and I've been directing the Center for Metabolic and Degenerative Diseases and running my lab for the past 15 years or so. We've been funded on a number of projects, and I'll talk about some of them here in this presentation slide. So basically, we are interested in various aspects of metabolic disease, obesity, fibrosis, cancer, stem cell biology, and aging. So there is a lot that we're engaged in. But in general, we use mouse and cell culture models of disease, and we also do translational research, clinical sample analysis. So just some of the projects. So we're looking at fat tissue biology, various types of adipocytes, brown and white adipocytes, how they store lipids and how they burn them. And we're looking at how fatty acids are metabolized and transported through various cells of fat tissue. And it's important to understand this processes because adipose tissue is engaged in interactions with other tissues and including malignant tissues. So cancer obesity link is something that we've been very actively involved in. And we've discovered that adipose tissue serves as a source of lipids for tumors and also of cells that are recruited from adipose tissue by carcinomas and engage as cancer-associated fibroblasts. So we've been looking at mechanisms of how transport of molecules and cells is regulated between the tissues. And more recently, we've gotten engaged in aging research, and we created humanized models of cell senescence by knocking out telomerase in stem cells of fat tissue, and we looked at how adipocyte progenitors run out as a result of replicative senescence and how that predisposes to type 2 diabetes development. We've been using advanced technologies for all of these projects, including single-cell RNA sequencing and lineage tracing. And most of our efforts have been focused on understanding the role of adipocyte progenitors. So we've discovered that there are several populations of stem cells that give rise to brown adipocytes and white adipocytes. And we've been looking at how those adipose stem cells differentiate and how they engage in normal adipose tissue biology and pathology. We learned that in addition to engaging in cancer progression, they're also recruited by fibrotic tissues and contribute to the population of fibroblasts in diseases such as muscular dystrophy. And we've been coming up with approaches on how to suppress their function in pathogenesis of various diseases. And finally, what our lab has expertise in is isolating experimental drugs and using them for experimental biology applications. So we've been screening combinatorial peptide libraries in animals and also in patients, and we've identified peptides that bind to differentially expressed cell surface receptors in various tissues and on various cell types. And we have peptides which bind to vascular endothelial cells, perivascular cells, cancer cells, and we've used them as imaging modalities, as probes to deliver non-invasive imaging, fluorophores, for instance. And also we're converting them into experimental drugs, and we've created peptide-based therapeutics, which deplete cell types of interest. So here I'm showing some data where we used a peptide which targets adipose stem cells, and we showed that we can suppress not only tumor growth, but also metastatic cancer progression. And through these exercises, we learned how adipose stem cells contribute to cancer progression. We showed that when we deplete them, we lose the important chemokine CXL12 that promotes the epithelial to mesenchymal transition, cancer metastasis, and chemotherapy resistance. And in addition to this peptide, we have a number of others that have been patented and have gone through clinical trials. So any students who are interested in these projects and join the lab would get exposure to all of these projects and will learn how to screen combinatorial peptide libraries and how to work in models of disease and cell culture, animal models, and translational research. Thank you.